Well, good afternoon and welcome to today's webinar looking at science and technology demonstrations launching on board Northrop Grumman's 21st Commercial Resupply Services mission to the International Space Station. I'm Patrick O'Neill, Public Affairs and Outreach Lead for the International Space Station National Laboratory, and I'll be your moderator for today's event. Prior to getting into some of the research investigations that are destined to the space station on this mission, let's talk about some of the logistics for this launch. So as mentioned, this is Northrop Grumman's 21st Commercial Resupply Services mission to the orbiting laboratory contracted by NASA. The launch is slated to take place from Space Launch Complex 40 in Cape Canaveral Space Force Station in Florida in early August. Northrop Grumman's Cygnus spacecraft is filled with more than 8,500 pounds of critical research and supplies and will launch on a SpaceX Falcon 9 rocket. Speaking of research, that is why we are here today, as we have an impressive lineup of individuals that are ready to talk about their investigations that are launching to the orbiting laboratory. These representatives will provide an overview of their projects and then answer any questions to media who have registered in advance. This webinar is also being recorded and will be posted to the ISS National Lab YouTube page, as well as our official NG-21 launch page. Okay, before we hear from the teams, though, launching investigations on this mission, we will first hear from a few representatives from the ISS National Laboratory, NASA's ISS program, and after which we will have some special remarks from the National Science Foundation. For media, after these opening remarks from ISS National Lab, NASA, and NSF are done, I will open it up to any questions. To ask a question, please raise your hand feature within the Zoom platform, and we will unmute your line so that you may ask your question, or you can just ask your question by sending it in via chat. Okay, so I think that should put us in a pretty good place as far as logistics are concerned. So what do you say we start talking about some science? First, I would like to invite Dr. Davide Marotta, Program Director in Space Biomanufacturing for the ISS National Laboratory, to provide some remarks from research that is flying from the ISS National Lab.
Good afternoon and happy Friday, everyone. Excited to be here with you all today as we talk about some of the research and technology development flying on Northrop Grumman's 21st Commercial Resupply Services mission to the space station. As Patrick alluded to, there are a variety of payloads flying on this mission that the astronauts will support over the coming weeks and months. But I'll let Dr. Everett speak to some of the projects NASA is sponsoring on this mission. And also, I don't want to step on the toes of some of the researchers who are here today to discuss their payloads. Instead, figured I would give some additional background on some of the other projects flying on the space station on NG-21, sponsored by the ISS National Lab. First and foremost, there is a tremendous diversity in payloads that are flying on this mission. We have live science projects, physical sciences, technology demonstrations, projects focused on inspiring the next generation, and more. So this will be a fantastic representation of the capabilities of the space station and the sort of research it can support. Shortly, we will have Dr. Susan Margulies from the US National Science Foundation providing an overview on their role in supporting fundamental research on the space station. And we will also have Dr. Alicia Boymelgreen, a researcher founded by the NSF on this mission, providing an overview on her team's investigation, but also on this mission are two other NSF-funded investigations, both of them looking at the physical sciences. On those projects stems from the University of Notre Dame, where this team will build on prior research funded by the NSF to study a fundamental physics process that uses light to generate bubbles that can collect, concentrate, and deposit trace amounts of substances in a solution on a surface for biosensor technology applications. As mentioned, this builds on prior research that used surface heating to generate bubbles with nanoscale patterns. The study looked at how the bubbles form, grow, and detach. But why this? Why? Because gravity-driven buoyancy is removed on board of the space station. And without buoyancy, the team can study what happens before bubbles form, which can lead to a variety of applications for us here on Earth. One of the major areas of strategic interest from both the ISS National Lab and NASA is in space production applications. On this mission, multiple projects selected through NASA's In Space Production Research Announcement of 2022 are flying under the sponsorship of the ISS National Lab. You will hear from Dr. Svendsen from Cedars-Sinai shortly, but a team from CU Boulder, together with BioServe Space Technologies, will use this funding opportunity to launch and validate a novel stem cell bioreactor nicknamed BICEP, which may provide a new technology for future spaceflight production of clinical-grade stem cells. With additional technologies like biceps, indeed, to manufacture, for example, hematopoietic stem cells in space, this could further expand in-space production application on this space station for the research community to leverage. We have spoken about this project previously, but Madison Square Garden Entertainment and Sphere Entertainment are partnering to launch their second interaction on the MSG Sphere camera. The technology demonstration aims to develop and test a new ultra-high resolution single sensor camera to capture groundbreaking imaging of space from the ISS. The MSG Sphere camera will provide high fidelity, unprecedented resolution, an extremely wide field of view, and a level of details and sharpness that is far beyond anything currently available commercially. The camera could enable Earth imaging in ways not previously possible, and the imagery could be used to create immersive experiences for viewers on Earth. That gives a bit of a snapshot of some of the projects launching to the space station sponsored by the ISS National Lab on this mission. I encourage everyone in the coming days to go to the ISS National Lab website, as we will have a series of releases, videos, and features on the projects launching on NG-21. And lastly, next week, I'd like to remind we have the kickoff of the 13th Annual International Space Station Research and Development Conference in Boston. This is the annual showcase of R&D in low Earth orbit and how the research and technology is fostering commerce in low Earth orbit and benefiting humanity. I'm sure that Patrick will talk a little more about the conference, but again, very excited about this launch. A lot of fantastic research and technology. Demonstrations are flying out of the Space Coast, and best of luck to our friends at Northrop Grumman and SpaceX on a successful mission, and go Cygnus.
Awesome. Thank you very much, Davide. Next up, I would like to welcome Dr. Meghan Everett. She is the Deputy Scientist for NASA's International Space Station program to provide a perspective from our nation's space agency.
Thank you, Patrick. So with all of our launches, I'm always happy to be here today to represent NASA with our other partners in science and with these brilliant scientists that we have to talk about their experiments that will be flying up on NG-21. So for this flight, it actually comes at a very busy time on Space Station because we will have crew handover right around the same time that NG-21 launches. So what that means is that we will have new crew members joining our residents on the space station, and then we will have crew members that have been living on the space station for a while. They'll be returning back to Earth. So we'll have a lot of human science during this time as we're handing over those crew and trying to get those first data points and those last data points as the crew arrive and they return home. So in addition to those great scientific discoveries that we'll be making on the human side, we are also flying new science on NG-21. In fact, we're flying 920 kilograms of science. So that is bringing a whole great new world of possibilities in science that we're going to be doing. We'll be fully utilizing our facilities on the International Space Station to make novel discoveries and to progress science beyond what we can do just on Earth by providing this microgravity environment where we can look at new things and our scientists can approach problems outside of the world of gravity. So some of the things that we look at on all of our rockets that launch to the International Space Station is we fly science that supports STEM research, fundamental sciences, so that's looking at physics and biological sciences, and how things react differently in the microgravity environment. We do technology demonstrations that both enhance humanity here on Earth, as well as promote our exploration beyond low Earth orbit to the lunar surface, or even looking beyond the lunar surface to Mars one day. And as Davide talked about, we also do in-space manufacturing. And we have great examples of all of that science flying on NG-21 today. We also have fantastic scientists that are going to talk some of it. So I'm going to try not to be redundant and talk about any of their science, but I'll talk about a couple of things that NASA is flying that we're really excited about, that will hopefully pursue our ability to explore beyond the International Space Station, as well as improve life here on Earth for folks. So one of those things is called the Packed Bed Reactor Water Recovery Series. And this specifically looks at two-phase flow of a liquid through different media. And this is actually the third flight for this piece of hardware. And the whole idea of flying it is how we better inform our water cleansing processes on the space station, as well as our urine processing on the space station. And as we're doing this, we want to be able to inform what type of materials are best for our engineers to design hardware for our future spacecraft. But this also is really important because it informs things on Earth, such as life support systems, fuel cells, nutrients to soils, or even how to better produce pharmaceutical grade water that would go into different drug developments. So we're hoping that this science really is informative on how we can get our systems ready to go beyond just the International Space Station for flights that we have just around the corner to the lunar surface. We are also looking at the effects of radiation on different life forms. So there's an investigation called Rotifer-2, and this is looking at DNA repair in rotifer, which are these kind of planktonic type organisms. So we'll be exposing these organisms to the harsh environment of microgravity and comparing those to the DNA of the organisms that are just here on Earth. And that should give us a great example of how long-term exposure to these radiations or these harsh environments affect all of our DNA processes, whether they're in humans or in other life forms. We will be doing a lot of work on on plant science, and both on our facilities as well as specific experiments that are going up that'll be discussed. And then another area that we're very keen on pushing forward is looking at different ways to cure diseases on the space station. One area that's been in the press and the media a lot is cancer moonshot, and NASA being a big partner in that project and that effort with the White House. And there are cell science and stem cell experiments that are specifically looking at how we can detect cancer earlier, how we can provide new and novel treatments for cancer and related to drug discovery or stem cell research, and even regenerative medicine that we will be talking about today as well. So with that, I am going to hand it back to Patrick and hear from the rest of our group today. Thank you.
Well, thank you very much, Dr. Everett. And now we are going to pass it over to a special guest. We have Dr. Susan Margulies, who is the Assistant Director of the National Science Foundation Directorate for Engineering, who will provide some remarks on how the NSF is providing funding to support fundamental research on the orbiting laboratory.
Thanks very much, Patrick. The U.S. National Science Foundation is an independent federal agency that supports science and engineering across all disciplines in all 50 states and U.S. territories. We fulfill our mission chiefly by making grants. We provide about 25% of federal funding to all the academic institutions in the U.S., colleges, universities. We fund basic research, and we also support solution-oriented research. We also support the training of the science, technology, engineering, and math workforce of tomorrow. K to gray, we like to say, from very early stage through college, through those who are already in the workforce, and reskilling for new careers. NSF invests in state-of-the-art facilities, test beds, and infrastructure to enable researchers across the US to explore new questions that they might not be able to explore on their own campuses, and to expand our understanding and test the designs of new technologies. The International Space Station is a very special test bed. The microgravity environment of the International Space Station allows researchers to study phenomena whose fundamental mechanisms might be masked by the effect of gravity. 3 projects that are going up in NG-21 will be investigating fluid dynamics, particulate processes, and nanoscale interactions to improve our understanding of problems here on Earth. Future technologies in biosensing, drug discovery, and environmental monitoring are going to arise from these experiments. NSF is a longstanding partner. We have been partnering with the ISS National Lab and NASA since 2015, and we have invested over $27 million in 80 awards. That's 58 different projects to 56 different institutions across our nation in 26 states for the fundamental research that takes place on the International Space Station. And more than 20 of those projects have been launched so far. Besides fluid dynamics, topics include behavior of heat and flame, as well as tissue engineering. So I wanted to highlight just a couple of really exciting projects that have yielded really important impacts so far. In the area of combustion engineering or fire, we've actually benefited from the studies in space to understand how fire moves in confined spaces. In particular, how it interacts both thermally, the temperature, as well as aerodynamically with the flow, and how fire spreads with thin walls and thick walls. We've literally changed how we fight fires and prevent fires to help reduce deaths and property damage from structural fires. In short, NSF invests in high-impact projects that have the potential to improve our understanding of life here on Earth. The engineering advances that we have funded address many, many challenges, including manufacturing materials in new and very different ways, treatments for diseases, clean energy innovations, and even flood and, of course, forest fire resilience as well. These are exciting projects that will really improve our understanding of phenomena on Earth and our ability to control and create new opportunities to improve life here on Earth. Thank you.
Fantastic. So at this point, we're starting to get to the point now where if there are any questions from the members of the media, again, you can either raise your hand within the Zoom feature, or you can ask your questions within the chat itself. But I did have a couple of questions actually for you, Dr. Margulies. So to your point, you mentioned that this is a longstanding relationship with the ISS National Lab. And while your team is funding an incredible amount of research, what is it about launching projects to the space station that is both exciting for you as well as for your team that, you know, kind of maybe is a little bit different about some of the other projects that you funded that you mentioned?
So space is a very special laboratory. I'm an experimentalist as well as a modeler, so I really embrace the purpose of the experiments that are done on Earth and the role that the experimental laboratory of the International Space Station plays. One of the projects I'm a biomedical engineer. One of the projects that I think has been especially exciting is we all know that there's muscle loss for our astronauts in space. But we also know that there's muscle loss for those of us on Earth, whether you have had a casted limb, whether you have family members who are aging, there's muscle loss. So a previous project that went up into the International Space Station explored in cells, is the muscle loss experienced in space the same as the muscle loss that's experienced here on Earth with aging or with disuse? So the answer is yes. So space becomes a wonderful accelerated test bed to be able to test new drugs that could combat muscle loss here on Earth. by testing new compounds in space. So that type of opportunity to really take science on Earth to space in a way that we could never study them on Earth is really exciting to me. Thank you.
Course. And you and I chatted a little bit before we came live with this, where you had the opportunity to go watch a launch recently over at Wallops Flight Facility out in Virginia. And, from your perspective, there has to be a sense of pride knowing that, there's science that are going on pretty much every single one of these resupply missions these days that would not have the opportunity to go to the space station had it not been for funding from the NSF. So can you maybe just talk about, like, again, what it is that you and your team feel knowing that every single time there's a rocket launch, your team has just a little bit of NSF investigations that are flying on that?
It's truly exciting. So the launch at Wallops was a really awesome experience. I had to go in the middle of the night. I had to meet at 1 o'clock in the morning. I was on a Trailways bus with a bunch of other congressional staffers. It was a lot of anticipation. But as we approached the town, you just had this whole sense of the town turning out for this event. And as we approach the Olympics today, we are, as a nation, so excited to see our teams compete. Science is also a team sport, but it's not a competitive team sport. It is a team sport that everybody can get behind and get excited about. The scientists and the engineers here on Earth, CASIS, the launch team, the astronauts in space who are our distant colleagues who are actually doing those experiments in space, and more. It's a local community, it's a global community that all celebrates science and engineering together. Truly awesome.
Fantastic. And yes, I can certainly attest that launches from that area are quite unique. So we look forward to hopefully going back there in the not too distant future. Again, thank you very much. I appreciate your thoughts and insight and looking forward to many more NSF investigations launching to the space station.
Thank you, Pat.
All right. So with that, now I'm going to move on to the research side of the house. So I would like to introduce first Dr. Alicia Boymelgreen from Florida International University. Dr. Boymelgreen and her team are launching an investigation funded by the NSF seeking to improve models of active colloids that could have a broad range of applications. So Dr. Boymelgreen, good afternoon, and please tell us a little more about your project.
Hi, so I am a assistant professor at Florida International University, and this project is one of those that is joint funded by the NSF and the International Space Station National Laboratory, and it's in collaboration with Dr. Jared Schiffbauer at Colorado Mesa University. And so I think I had some slides prepared if we can upload them, because it's just easier for me to explain things with pictures. So my research, I'm a mechanical engineer, but my research is in this field called active matter, which kind of traverses across multiple disciplines from physics to biology to material science to chemical engineering, mechanical engineering. We have a little footprint in all of these. But in a general, broad formulation, active matter is a type of system where objects draw energy from their surroundings and convert it into mechanical motion or movement. And so we most likely to recognize this in biological systems such as birds and fish and even bacteria. So it exists across all different length scales from very small to very big. But one of the defining characteristics of active matter is that if we look at a single active object when it's isolated and nobody's around, it will wander off on its own path in its own directions. But when we bring a whole bunch of active objects together, they create they interact with each other and produce patterns and formations that we refer to as collective motion. And examples of collective motion that are most familiar to us are things like flocks of birds or schools of fish, swarms of bacteria, things like that. So my research, next slide, is actually not in biology. I'm a mechanical engineer and I mainly deal with synthetic or artificial things. So we use synthetic and artificial kind of interchangeably. So we try and create these systems out of non-biological systems, out of artificial objects. And to do that, we fabricate these special particles or colloids that are microns in size, which if you want to scale is about a 50th of your hair, the thickness of a single hair. And we call these particles Janus particles because they have two different properties in the same object. And so they're named after the Roman god Janus, who was also the source of the word January. The Roman god Janus had two faces, as you can see in this picture here. And our particles also have two faces. So in my research, I work with electric field-driven active matter. And so for us, it's beneficial to have the two faces out of two different materials with different electrical properties. So we have an insulator, polystyrene, and we have a metal, gold. And what happens is that when we put these particles in a fluid, in water or in electrolyte, or our space experiments are actually going to be in glycerol, and then we apply an electric field, they wander off. If the solution is dilute, so there's not a lot of particles, they wander off all in different directions. And so on the left-hand side, you see a microscope image, and that's been tracked with a particle tracking algorithm, where the particles are running off in all different directions. They're not moving in one specific direction. And on the right-hand side, we have passive colloids that are not really moving at all. They jiggle around a little bit on the spot under what we call Brownian motion. So in order to understand why we want to take these particles to space, it's helpful to understand what doing experiments looks like on Earth and what the state of the research is on Earth in our labs and in the current research community. And this, next slide. This, basically, we have what I call a spherical cow or spherical chicken problem, which is a reference to this super nerdy physics joke where there's a farmer who his cow is not, depending which version of the joke you have, his cow is not producing milk or the chicken's not producing eggs, and he can't figure out why this is going on. So he calls up his friend, a physicist, and he says, you know, you're a really smart person. Can you help me out with my problem? And the physicist says, sure, I can solve it. And he goes away for 3 weeks, and he comes back to the farmer. He says, great news, I've solved your problem. The only issue is, the slight issue, it's only valid for a spherical cow in a vacuum. Now, this joke is funny because spherical cows don't exist, right? Cows are cow-shaped, and they don't generally live in a vacuum-like space. So the physicist came up with a problem that was easy for them to model, but it doesn't actually correlate with reality. And we have that a little bit in our active colloid research, where we derive theoretical models for a single particle that's floating around in the bulk. There's nobody around it, no walls around it. And then we do experiments. And what happens in the experiments is that we have this metallic coating. The metal is heavy, and so the particles drop down to the bottom of the chamber that we're looking at them in, and they move along near the wall. Now, if you're moving near the wall, you're interacting with the wall, and it kind of complicates the physics in a way that if anyone's a swimmer, you know, if you swim along at the bottom of the pool or if you swim somewhere near the top, it feels different. And the reason for this is because near the bottom of the pool, the wall is actually pushing against you and increasing the drag or the friction, and it's harder to swim against it. So what happens is we have theoretical models that are derived for single particles with no complications. And then we have experiments where all the particles are near the wall, which is super complicated. So then the experiments and the theory don't match. And we go back to the theory and we say, oh, the problem must have been the wall. So we're going to compensate for the wall. But we never actually checked the particle that is by itself. We never actually checked the theory on its own. So next slide. Our goal with microgravity is to make our particles float. So we're going to take the system on Earth that is what we call quasi two-dimensional because it's lying in one layer near the bottom of the chamber because all those particles have dropped down due to gravity and sedimentation. And we put it in space and all the particles are going to float. And we can take measurements now far away from the channel wall. And we expect that this will inform our understandings of the fundamental behavior of active matter. So for me as a mechanical engineer with a little bit of physicist bent, this is super interesting from a fundamental perspective, being able to validate theoretical models. But it's not the only thing that we can do. There's been a lot of talk in the active matter community, and this is my last slide, on what can we do with active systems. And there have been a whole lot of different applications proposed, ranging from targeted drug delivery. So can I deliver a drug to a specific cell that I want to deliver it to without having to deliver it throughout the whole body and have it diffuse through the whole body? My former advisor in Israel is working on single cell gene transfection. So can I change the gene inside a single cell without changing it in all the surrounding cells, and then look at what effect that has? We can also create new materials where we stack up different types of colloids all together and control them and move them with the electric fields. And then lastly, there has been some talk about using it for environmental remediation, including, so imagine that I have an oil spill and I put in these particles that can absorb oil, and they run around throughout the oil spill absorbing the oil. And yes, and finally, we also have applications to advanced manufacturing and self-repairing materials, and it's pretty endless. They're really rich systems that are super interdisciplinary, traversing multiple areas of research and application. And so we're really excited to see what happens in space, how the particles behave, to understand their fundamental driving mechanisms, and then to take that understanding and apply it to real world situations and commercial applications. Thank you.
Awesome. Well, thank you, Dr. Boymelgreen. Again, if there are any questions from media, please let us know. But I also like the swimming analogy. That's very appropriate given what's going on with the Olympics and getting those fired up later on today, hopefully. So question on my end too is, I believe that you're working with Space Tango on this particular project. Can you maybe talk a little bit about how this project is going to be I guess Space Tango has autonomous platforms on the space station, so it's not necessarily going to be the astronauts that are going to be actively working on them. Maybe talk a little bit about how you configured this project within this autonomous platform.
Yes, thank you. So Space Tango is what we call our implementation partner. So their job is to interface between us, the scientists who don't know too much about space, and the people on the International Space Station, and to translate our science to make it space ready and kind of alleviate that process and make it more efficient. The unique aspect of Space Tango's technology is that their systems are automated. So when we originally considered this research, we had two options. One would be to send our experiments to space and hand them over to the astronauts, who would then put them under a microscope, apply the electric field, and do the actual experiments. And the other option, which is what Space Tango provide, is it's like an experiment in a box where we are going to load our microfluidic chips, or the chips, the system containing the active colloids, into an automated platform with its own microscope, its own electric field generator. And then we actually Space Tango can control it from Earth if we need to make any modifications. So there's this real-time ability to alter things if we need. But essentially, everything is pre-programmed to have the microscope change the range of visualization, so it can go up and down and look at different layers inside our active colloid system. And the electric field runs on a specific algorithm to change the voltage and the frequency of the applied field. And so our system is fully automated, and this has a big advantage that it doesn't require any time of the astronauts, and makes it a little more easier on them, and could be more economically feasible to scale up and things like that. So yeah, we've been working with Space Tango. It's been really great. We're excited to see how it works. We ran through what we call a payload verification test, where we checked that everything was working and running and that our chips were behaving and we could visualize everything. A big challenge was figuring out the visualization that would be going from 2-dimensional on Earth to 3-dimensional. And yeah, that I hope that answered the question a little.
That absolutely did. And again, I think that it's always interesting to hear how it is that our research teams are taking something from that they're very familiar with trying to do on Earth, but then trying to conceptualize that and turn that into a project that will launch into a very constrained environment on the space station.
Yes.
So thank you very much. That was great.
Yes, I would have liked to squeeze my graduate student inside the box just to have a backup, but apparently we're not there yet with the technology.
I'm sure they would have enjoyed the ride too. So. Well, thank you very much, Dr. Boymelgreen, and we look forward to hearing more about your investigation as it launches in the coming days and weeks. With that, I would like to move on to Dr. Tony Atala. Dr. Tony Atala is, I'm sorry, hold on real quick. He is with the Wake Forest Institute for Regenerative Medicine, and they are launching an investigation that is going to be evaluating 3D printed liver tissues in the novel environment of space. So Dr. Atala, please provide some insight into this project.
Thank you so much, Patrick. It's really great to be with you today and really great to be with all of you. We are involved in the area of regenerative medicine, which is really an area that uses the patient's own cells to create cell therapies, tissues, and organs. We can do this for a number of tissues that were already implanted into patients. So our partners in this endeavor for this particular project, of course, is NASA, the ISS National Laboratories, and Redwire, who have enabled our experiments in this mission. Basically, this comes out of the NASA Vascular Challenge. NASA put out a challenge a few years ago, and that involved creating a vascularized solid tissue that would remain functional outside the body for over 1 month. And this, in fact, was quite a challenge, because when we create these tissues, we try to implant them right back into the in vivo situation where they will be able to function normally. But to keep them alive outside the body for a month was quite a challenge. And so we were lucky enough to have been able to achieve that challenge by using a strategy of 3D printing. And we did this by targeting liver tissue. And so liver is a very complex organ, very vascular, very, very vascular, large mass. And what you can see here in this slide is this vascular tree that is present, that is extremely complex, but the tissue architecture is that of a gyroid. So that's the approach that we took in creating these structures, by creating gyroid blocks of tissue that we could print. And this is on the bottom, you can see a simulation of these blocks. And of course, this required a lot of cell work with cell expansion and the ability to print these with the cells into a structure that would have this gyroid appearance that is exactly what the liver structure is. So by replicating what nature does, we hypothesized that by doing so, we could achieve what nature is able to achieve, which is functional solid tissue that could, in fact, remain viable outside the body. And so this on the far left, you see this gyroid structure that we created through 3D printing. In the second middle panel, you see this fully seated with the cells. And then we were able to achieve that functionality long-term with these vascular channels that were able to maintain the tissue alive and functional. And now the next step for this, after getting this challenge and being able to achieve this challenge with NASA, then we were able to go to the next step with further funding from NASA and the ISSNL with our partner Redwire to actually create these bioreactors that would allow us now to do the same thing in space and to be able to take this to the space station for 30 days. Now, this in itself was yet an additional challenge because we had to make sure that these systems were perfused in these anti-gravity situations. And this was actually a favorable thing for us because on Earth, when we seed these structures and print these structures, gravity does not allow the cells to be necessarily evenly distributed. So there are actually major advantages to doing this in space, because in the gravity conditions of space, you do have the ability for these cells to be evenly distributed. And that is what we're looking at through these experiments by sending these perfused chambers with these bioprinted 3D liver structures into space, into the space station, and where we will be able to determine over a 30-day period exactly how these vascular channels form, how the cells get distributed, and this information will be critical for us to take next steps here with this technology on Earth. So we're going to be analyzing this 10 days, 20 days, 30 days. As these structures get preserved, we're able to bring them back to Earth, at which point we'll be able to analyze them at every level, where we're able to do genomics, transcriptomics, proteomics, metabolomics, structural analysis, to really try to understand the best way possible how this microgravity environment is able to help these things in space in terms of the formation of functional tissue that it will eventually help us here on Earth. Thank you so much for your attention.
Fantastic, Dr. Atala. So one of the questions that you brought up, that this came originally from the NASA Vascular Tissue Challenge, and when your team was first working on this challenge, was the thought that this would eventually turn into a space project, or was that kind of a happy accident that materialized as things moved forward?
Well, it's interesting. We knew that the project, of course, was to be able to create these structures on Earth, and of course, the actual price was to be able to get this funded so we could take this mission into space. So that was really a great thing for us to be able to do that. And we had 2 projects actually in the Vascular Challenge. We were lucky enough to have both projects succeed. So we got funded for both, and we're very, very excited to be part of this NG-21 to take our first tissue up into space through this Vascular Challenge component.
Well, we are excited to talk about it as well. And then, I think that also is we're looking down the road, and I had the chance to go and meet with yourself as well as Dr. Yu a couple of weeks back over at your facilities at Wake Forest. It's kind of looking at in-space production applications, using that space-based environment to possibly, create more, possible, like either organs or tissues, things like that could eventually be given to patients here on Earth. I mean, what's kind of exciting to you as you start to look at the future of how space might be able to influence biomedical research?
Well, just the ability to create tissues and organs, of course, has been a major advance for us in the field. And the question is, how can we really help to accelerate that technology? And, we have an amazing team here at W Firm. You mentioned Dr. Yu, Dr. Chelsea Wilson, and an amazing team of individuals all working together to bring this technologies really from the bench to the bedside. But this element of space is actually giving us this extra dimension that will help us to manufacture these tissues better here on Earth, and will allow us to have better conditions, we believe, that will help to lower cost, scale up the technologies, and accelerate the delivery of these technologies here on Earth, and hopefully eventually also in space.
Fantastic. Well, again, we look forward to hearing more about your investigation as it launches, as well as future investigations from your team. So thank you very much. And with that, I'm going to pass it now over to Dr. Clive Svendsen, who is with the Cedars-Sinai Medical Center. Over the years, Dr. Svendsen and his team have launched a variety of investigations to the space station, but this project is looking at in-space manufacturing of stem cells to improve regenerative medicine for patients on Earth. Dr. Svendsen, please tell us a little bit more about your project.
Wait, can you hear me okay? I'll just bring the slides up. That would be great. I'm here with Arun Sharma, who's my colleague. Can I have the next slide? In this work, along with his team. So induced pluripotent stem cells are an amazing technology that has come around recently. You can take adult skin cells from any patient, reprogram them back in time to what's called a pluripotent state. From there, you can make any tissue of the human body. And so we're thinking about using these cells for transplanting into patients. The next slide. But we can also use it for what we call disease modeling, making organoids in the dish. And in a sense, you're taking cells back in time, rather like this H.G. Wells story. So we can take an adult cell and send it all the way back to the beginning, and then replay the development of those cells in the tissue culture dish. Now, the next slide. So we're very excited about this technology, and Arun and I wrote a review on this recently. And there's many different aspects of stem cells that we can utilize in addition to cell therapies. and actually, you can take them down different routes. And many of these ideas have been used actually in the space station already, organ-on-chip technologies, making organoids from these pluripotent cells. And on Earth here, we're even transplanting these cells back into animals to see how they can function, as Tony was mentioning, for a cell therapy approach. The next slide. But one of the ideas that we had, Arun and I, some time ago, is two things really, and this is also with our colleague Drew Serene here at Cedars-Sinai, who with our partners at the hospital, we're trying to manufacture these cells and grow them at scale. And the challenges with these pluripotent cells is, in this graph here shows, to remain pluripotent, they kind of have to sit on a top and be very, very non-differentiated. So they're very, what we call naive pluripotency. However, over time, they start differentiating, and we can't really control that, and they push down to the different organ tissues. And we think gravity may have a role in this. So the next one. The two major questions we've asked is, I think you want to go back. Sorry, that's it. Two questions. Does microgravity keep iPS cells in a really naive pluripotent state, which would be very good for the field because it's hard to do that? And then if we do start differentiating them, how does microgravity affect the ability to make brain organoids, heart tissues, and eye tissues, and maybe even to the point where we can make whole organs in space? That might be an advantage to not having gravity in that situation. Those are the kind of things we want to test. Next one. So this is a team, and again, Arun's here. We have Maida, and Drew Serene, and another person who's joining Cedars-Sinai, Sonia Shepner. And together we've done a lot of work already with Space Tango. We just heard about that as one of our partners. Currently we have a mission coming up. If you just tap it again, you'll put a circle around this Axiom 1 mission. This is another grant that hopefully we'll be hearing about very soon from Arun, from NASA as well. The thing I'm going to talk about for a few seconds now is the current mission, which we're going up on, which is NG-21. So the next slide. And this is part of the INSPIRE program, and we're very happy for our partners NASA for funding this. The next slide, please. And it's in collaboration with BioServe, who's our partner in this case. We use Space Tango for other projects, but in this case, it's BioServe. And we're also doing this very much in collaboration with Axiom, who's another partner in this mission. So we're going to take fibroblasts. These are adult cells from patients and also from a special line that goes green when it gets pluripotent. We're going to transfect them with reprogramming factors. Next slide. And you'll see that when you reprogram these cells, they light up green because we've got a gene in it that shows green when the cells become pluripotent. Now, the next slide. We had a preliminary mission using this technology and showed that the technology of actually transfecting, putting genes into these cells in microgravity can work. And we got some nice red cells here using a red fluorescent protein. And you can see alongside those red cells are the pluripotent cells. So that's one of the experiments is, can we do for the first time, make these cells in space? So take an adult cell, reprogram it, send it back in time in microgravity. And is that more efficient? Does it work better? And also, will they grow better? Maybe they'll maintain their pluripotency at a higher level when they're grown in microgravity. The next slide. So we're very excited to launch this. If you want to see more about the work, we had this nice review recently. The next slide. It describes some of the things I just talked about. Next slide. And the bigger picture of how we can use the space station and microgravity to understand not just the cells that we're growing now and iPS cells, but cardiomyocytes, neurons, bone. We heard about muscle degeneration. We can actually make muscle cells from iPS and then understand, as we heard earlier on, how they are affected at the molecular level by microgravity. And that might relate, as we heard earlier, to human diseases. And finally, we can do things like astronaut health. We can think about monitoring the astronauts, making their own iPS cells and seeing how they are affected by space travel in order to understand what they might expect when they go up to the space station or when they travel in microgravity. In addition to, ultimately, maybe we can do drug screening if those models are very powerful, give us information that we can't get on Earth. And then finally, just, I think generally doing these experiments and having had the chance to work a lot on this, it really pushes you to think about how to grow cells in different ways and automate your technology by knowing that you've got to limit your time with the astronauts and do things in a much more compact way. Last slide. These are our partners. We thank everybody very much. particularly Janet Studemeyer, who's a partner with us, Drew, and all the folks on the slide. Thanks very much.
Awesome. Well, thank you very much, Dr. Svendsen. And, as I'm listening to your presentation, and also as I was listening to Dr. Atala's as well, it kind of makes me think about the fact that, we think of the world of like science fiction, but then I also think about a lot of the research that's happening on the space station right now, and it almost kind of makes it seem as though we're morphing those two, where it's almost like the world of science fiction and the world of science reality and now is coming closer and closer. Is that something that maybe you would agree with?
Yeah, for sure. I mean, I think the thought of being able to grow these cells under these conditions is amazing. I just think the actual idea of this, what we call induced pluripotent stem cells, the fact you can take a 100-year-old patient, take a cell and put it all the way back to an embryonic state is remarkable in itself. And if we can capture that and enhance the process using microgravity, I think that's going to be very exciting, and we will lead to this kind of era of perhaps making organs, having a whole, satellite going around Earth making organs. Now, a bit scary, does sound like a science fiction movie, but we're doing it, we're a hospital, we're doing this with approvals, with consents, do it the proper way. And the idea is this might impact patient health in the future. And we always joke that, Cedars-Sinai, we're the hospital to the stars here in this area of Hollywood, but we want to be the hospital going to the stars and helping people reach the new heights. Sorry about that.
I like that.
But also help explore the universe in the future, be the hospital of the universe.
Well, so into that end, I mean, it's almost like using space in some ways to go out there and further influence personalized medicine for people on the ground. Is that accurate?
Yeah, definitely. I mean, I think ultimately we're going to learn so much, as we heard from the previous speakers. We also have interesting things going on here. Brennan Spiegel is working on the gut and how the gut reacts to gravity on Earth, and how reducing gravity can be good for some diseases of the gut, inflammatory bowel disease, because of the weight is lifted. So we can learn a lot, I think, in medicine from understanding what goes on in microgravity, maybe applying some of that to clinics on Earth that we would be surprised about. And for the muscle discussion, the opposite direction, it's bad for you. But so how do you correct that on Earth? And understanding more using space station is exciting for medicine.
Perfect. Well, again, I know that I'm going to see Arun next week, but hopefully I'll get a chance to go out there and spend some time with you at a forthcoming rocket launch, because again, it seems like your team is doing quite a bit these days, launching stuff to the space station. So continued success.
Thank you.
Awesome. All righty. So next, we are going to be talking with Michelle Hooks. She is the Education Project Manager at NASA's Office of STEM and Engagement, who will talk to us today about a fun STEMonstration that will be launching to the space station. Michelle?
Hello, everyone. I am so excited to be here and to share our upcoming Screaming Balloons demonstration. I'm Michelle Hooks, an Education Project Manager on the InSTEM contract for NASA's Office of STEM Engagement. I support the Next Gen STEM project that focuses on specifically connecting kindergarten through 12th grade students with educational opportunities that are aligned to NASA's missions. Demonstrations really look at highlighting NASA's missions and work on the International Space Station. They're one project in our NGS portfolio for kids and teachers to connect with the science that's happening on station. Launching on NG-21, we sent several low-cost, fairly accessible resources that nearly any student can use to do an experiment with an astronaut aboard the space station. Demonstrations are one of these many projects that we have on NGS portfolio that aim to immerse students in NASA's work, enhance STEM literacy, and inspire the next generation to explore. So as I'm listening to all these different research projects that are happening on station and that are also launching on NG-21, my ears are already buzzing, and I'm thinking about ways that we can engage students further with some of this other research that's happening or that will be launching on NG-21. Filmed by astronauts living and working on the space stations, demonstrations utilize that unique microgravity environment to demonstrate critical concepts in K-12 education in short 3 to 5 minute videos. So they're quick, they're short, and they're full of graphics and other visual things to enhance student learning and to make them engaging for students. But the star of the show really are our astronauts as they demonstrate these concepts in microgravity in a way that you just can't possibly demonstrate in a classroom. Some of the topics covered include photosynthesis, Newton's laws, properties of water, and space art. So we have a really wide range of topics that we're covering and looking at different avenues that we can connect students to what's happening on station. The short videos also connect what students are learning in school to the ongoing research on station, and they explain how those concepts have implications for life on Earth, as well as the future of human space exploration. They give students kind of like an inside look at what astronauts are working on in space and highlight some of that research and technological advancements that are happening. For example, just this week, we had the 3D printing demonstration that just filmed on station. We're excited to get that released for students and educators. And last week, we had a demonstration that filmed that highlighted genetic research happening on station. To do this, we collaborate with strategic partners, with researchers, with engineers, astronauts, among others, to collaboratively answer that question that so many students ask as they're learning in school. And that's, why do I need to learn this? Why does it matter? How's it relevant in real life? And we're intentional about making career connections in each demonstration so that students can connect the dots between what they're learning in class, what the astronauts are working on in space, and then the many careers that are contributing to in various capacities to this work. Our growing library of demonstrations includes over 30 demonstrations that have been very well received by both students and educators. And we love getting pictures and photos and kind of feedback from students around the country and also around the world that are engaging as some of our international astronauts participate in this demonstrations in various capacities. For our screaming balloon demonstration, we wanted to involve students around the country in a unique student-friendly experiment with the astronauts that are happening on station. So to make this accessible to as many students as possible, we launched on are launching on NG-21 several supplies. We have balloons, hex nuts, pennies, and a balloon pump. We sent clear balloons to space so students can see what's happening to the coins that are going to be placed inside the balloons, and students on Earth will get to use the same supplies at an in-person event that streams online, so kids around the country can also participate. And we'll use these same supplies in classrooms on Earth, of course, as well as on station, and compare how that unique microgravity on station is impacting things. Astronauts on the orbiting library will place that penny in one balloon and a hex nut in a second, and they'll use the balloon pump to inflate the balloons. Then they'll give each balloon a gentle whirl to set the object inside in motion. Students will follow along with the astronauts and then compare what they see happening with their balloons to what's happening with the balloons that are in space. After giving the balloons a gentle whirl, the astronaut and students will let go of their balloon, and we'll see how our results compare. To really drive this point home and to complement this demonstration, the crew on station will highlight and explain some of the science behind this screaming balloon demonstration that students are doing, and they'll listen also, and they'll hear a certain noise as they're doing this, and that gives that screaming sound. And we'll see, how does that sound different, on space, or on station versus in our classrooms. And some of the questions that the astronauts will kind of probe will be, what keeps the penny going? How does the hex nut interact differently with this balloon as it's moving? What are some of the differences? And the astronauts will help students understand the role centripetal force is playing both in this screaming balloon demo, as well as in keeping the International Space Station in orbit around the Earth. Another force at work here that we'll explore is friction, and astronauts will explain that and talk about that in depth. And the biggest force, of course, that's causing a difference in our two experiments is going to be that microgravity environment, and that's the big spark and wow factor that we're looking to really highlight for our students. One of the major hazards we had to mitigate during planning for this demonstration was how to get the balloons deflated without popping. That was a safety hazard that was brought up, and we had to control for this. And so we have a controlled deflation that was sort of planned for But before the students see this, students will have the supplies on hand to try to deflate their own balloons without popping them. And then they'll watch as the balloons on station are deflated by the astronauts. And I can tell you, I tried this with about 100 students here in Houston, and they came up with all sorts of ideas to successfully and unsuccessfully either pop or deflate these balloons. And that was really, really fascinating for them to learn about some of the constraints and things that we face when astronauts are doing experiments, research, or technological demonstrations in space, like that there are legit and real concerns and safety hazards that need to be mitigated. So we've been documenting some of that behind the scenes journey that we've had to deal with and learn about, so that we can share that with students as well and give them kind of an insight look of like, hey, here's what's happening. Here's what, some of the concerns and constraints are. Like all of our demonstrations, the Screaming Balloons demonstration will have a corresponding standards-aligned classroom lesson plan that educators can use in their classrooms to dive deeper with their students in class, and really use this not just as an exciting and engaging activity, but something that really moves their curriculum and student learning forward. And I think that's about it.
Okay, and Tim, I think that there was an image or a series of images, I guess, that Michelle had as well. I don't know if you wanted to speak to some of those by chance.
Sure. And these are some, we were just capturing kind of pictures along the journey to share and to give students a behind the scenes look at what is happening. In the center, you will see the balloon with our pennies and the hex nut that are getting packaged. And we had to have them do kind of like a stowage check to make sure that they're going to be in the right configuration. On the bottom left is us with our HFIT representative that did the entire check to make sure that this configuration worked, and gave us that final human factors implementation evaluation and certificate of compliance. And then on the right are those RFID labels that were attached to each of these Ziploc bags or bags, so that we could track these supplies while they're on station.
Fantastic. And then question on my end, first of all, I certainly understand the joys of safety hazards and trying to mitigate those when you're launching something to the space station. But you also mentioned that there have been more than 30 different demonstrations that had taken place thus far. I mean, you mentioned another one of them, but are there some other examples that you could provide that kind of give an insight or an understanding of like the full breadth of demonstrations that are available for people to learn more about?
Yes, absolutely. And most of our demonstrations, we rely on items that are on station, and we look at including items that students can kind of get their hands on. So for example, properties of water, we used, it was like a hygiene cloth, so basically like a towel, and water. And we saw that get soaked with water, and then we watched as the astronaut, gave it a squeeze, and then saw what happened in space, and students could try the same thing in their classrooms. And we have several just like that utilize either simple supplies that are on station, or the astronauts will use their bodies themselves to either move around to highlight things like friction, or other topics on station.
Very cool. And since these are very visually, focused, where's the best place for students and teachers alike to go and learn more about these?
Yes, they're available on nasa.gov and also streaming on NASA Plus and our YouTube channel, the NASA STEM YouTube channel.
Awesome. Well, thank you very much, Michelle. And also, I love the background too. That is very, very spacey. Okay, well then, last but not least, we have Dr. Pabudu Handekumbura, team biology leader at the Pacific Northwest National Laboratory, to discuss APEX-09, which examines carbon dioxide capture mechanisms of two types of gases, which I will certainly not try and pronounce right now. So I will defer to you to be able to pronounce those two gases. So thank you so much.
Thank you. I'm excited to be here today and share a little bit about the work we do. So like Patrick mentioned, I am Pabudu Handekumbura. I'm a plant biologist and a team leader at the Pacific Northwest National Laboratory. I'm also the principal investigator for the C4 photosynthesis in space project. So plants are remarkably capable of capturing the atmospheric carbon dioxide and then converting it into oxygen and sugars, a process called photosynthesis. Certain grasses like maize and sorghum have more efficient photosynthesis mechanisms, which we call the C4 photosynthesis mechanisms, compared to some of the other grasses like rice, which has a C3 mechanism. So how this carbon dioxide concentrating mechanism is changing in microgravity, and how is the C3 and C4 grasses differing in microgravity environment is the investigation of our project. So for this, we are using two model grass species. One of the species, a C3 model called Brachypodium distachyon, and a C4 model grass species that's widely used called Sitaria viridis. So these, we have set up our experiment so that we have the space station conditions, ISS conditions, and then we have two ground controls, so that we can try and understand how the photosynthesis and the overall carbon metabolism of these species are changing in microgravity compared to the conditions that we have on Earth. And with this investigation, we are hoping we'll be able to broaden our understanding of how plants respond to various stresses, both on Earth as well as on the space station, where there is extreme levels of carbon dioxide and microgravity. And we are hoping that this will enable us to help design future plant-based systems for bioregenerative support, as we are thinking of deep space exploration and how to use plants both for food and for aesthetic purposes in those space explorations. So I think that's briefly what we are hoping to accomplish with this experiment, and I'm very excited for the launch, this experiment in NG-21.
Fantastic. Oops, excuse me. Down the wrong tube for a second. Sorry about that. Question on my end then too. So you mentioned that there's the two species that are flying on this mission. Do they offer advantages over smaller types of plants? Maybe explain kind of why it was that these species were selected.
Yeah. So now broadly, there's two types of photosynthetic carbon capture mechanisms, the C3 and C4, right? So I picked two species. One had a C3, one had a C4 carbon capture mechanism. And these two grasses, Brachypodium and Sitaria, are widely used by the scientific community to study. They are model grasses, a model system that we use and study and infer based on what we find. So these are both small stature grasses that can fit within the, so like the image that's shown here, this is set up for growing these plants in the two veggie units that are available in the International Space Station. So it's kind of like a growth chamber where you have the lights, you can control the daylight cycle, similar to what we usually have in our growth chambers, where you can program your day-night cycle and the different parameters you want to test out. So both of these grasses, they can grow and fit within these space limitations we have. And then they are ideal models, widely used by the scientific community. So that was the reason for choosing Brachypodium and Sitaria. A lot is known and a lot of research has been done on these two species. But for Sitaria, the C4 grass, that has never been flown to the space station before. So that would be a first. And we'll learn more by sending it and just growing it out on the space station conditions, how to establish them, how to because this experiment is about a 33-day long experiment. So we'll be able to capture a few different developmental stages for both of these plant species. So that'll help us as we think about the effects of microgravity and the different stresses that plants, same as the humans, plants need to experience and cope with in these extraterrestrial environments.
Very good. A question on my end too is, given the fact that you work for a national laboratory right now, it's probably has to be pretty interesting to know that you're able to do your research within your national laboratory, but then now you're also launching something to the International Space Station. I mean, maybe talk about like the juxtaposition of how that all came to be in the first place.
Yeah, this actually is a really great opportunity. So this work is funded through NASA. Just like any other like NSF or DOE. So this was a funding opportunity that came up, and then we were lucky to like get selected to do this work and to really try this unique microgravity environment out at the space station. So yes, I am at a national lab where we have access to world-class instrumentation and capabilities. Especially where I am at, I am coming also from the Environmental Molecular Sciences Laboratory, which is a user facility run by the Department of Energy. So we actually have so many world-class cool capabilities within our facility. So with the funding I got from NASA, now we are able to really explore the microgravity component and how those multiple stresses impacts plant performance, photosynthesis, and overall plant metabolism. And using the omics capabilities we have at the National Lab, we are able to generate multi-omics data sets that can be integrated into different modeling platforms. And then the contrast we have between gravity and the microgravity, I think that will open up so many opportunities for the research community. I think this would be I think I'm most excited about the opportunities that this will open up and the opportunity to broaden our understanding of the fundamental sciences, how species, how humans, plants, organisms interact with the various stresses. And what changes internally. So yes, so it's a unique opportunity to be able to leverage some of the capabilities we have, as well as collaborate with NASA.
Awesome. Yeah. So it's almost like, I kind of liken it to where you live in a 3D world. Now, all of a sudden you have the opportunity to be add a fourth dimension to it. What kind of things can you learn or extrapolate from that when you have this new dimension that's at your disposal? So again, very excited. And I know that the NASA team is equally excited about your investigation that's launching on this one. And hopefully that is the first of many from your team.
I hope so.
Awesome. Well, at this point, again, if there are any questions from media, please feel free to either raise your hand, ask them in the Q&A section, and we'll kind of give it a second. If not, then we'll get ready to wrap things up. Okay, well then, if that is the case, I would like to thank all of our panelists. I would like to thank those that also joined us from the media today to learn a little bit more about some of the projects that are launching on Northrop Grumman's 21st Commercial Resupply Services mission to the International Space Station. As mentioned, we are looking at launch in early August from Cape Canaveral Space Force Station in Florida. For up-to-the-minute information, please follow @NASA or @NASA Kennedy via social platforms, or follow the coverage on NASA's International Space Station blog. But before I do go, Davide mentioned this a little bit earlier today. I have to throw in one final plug for the 13th Annual International Space Station Research and Development Conference that is going to be taking place next week in Boston, Massachusetts. For media that are not going to be able to attend in person, the plenary sessions will all be on a live stream, which is going to be available at issconference.org. So we look forward to seeing a lot of our research partners out in Boston next week as we learn about the great things that are happening on the space station, as well as great things that will be happening in the future in low Earth orbit. Okay, so again, we hope that you found today to be very informative. With that, it's been a pleasure to moderate today's webinar. On behalf of the ISS National Lab, I am Patrick O'Neill. Go Dragon, go Cygnus, and most importantly, go space and go science. Have a good day and have a great weekend.